بسم الله الرحمن الرحيم. Neuroanatomy by Dr. Ayman Ahmad Khamfour, Professor of Anatomy and Embryology, Alexandria University, Egypt. Development of the CNS. It will include formation of the neural tube and neural crest, development of the spinal cord, development of the medulla, and show in between a group of short questions. Now we will start by development of the neural tube. What we see now is just the surface ectoderm of the developing embryo. At the third week intrauterine fetal life, there is a thickening of the surface ectoderm in the midline of the embryo in the region overlying the notochord, forming what's called the neural plate. This plate is deepened to form the neural groove. Meanwhile, this neural groove closes to form the neural tube, which closes at the fourth week intrauterine fetal life. So again, we have neural plate, neural groove, and neural tube. Look at the edges of the neural groove. We find that it is markedly thickened, forming the neural crest cells. These neural crest cells becomes separated and detached from the surface ectoderm and lies on each side of the developing neural tube. The neural tube will form the future brain and spinal cord, while the neural canal will form the cavities of the brain, which are the ventricles, and the central canal of the spinal cord. This is the neural groove, which starts closing in its middle part and proceed more upwards and downwards. But it leaves two openings at the cephalic region and in the caudal region. They are called anterior neuropore and posterior neuropore. There's another diagram showing the formation of the neural groove, neural tube, and neural crest cells. The same idea with closure of the neural groove to form the neural tube. This is a very nice animation showing how the neural groove is formed and how it proceeds to form the neural tube and how the neural crests are formed. Please, in this animation, concentrate on two colors: the light blue color and the green color. Again, there's a light blue color, groove, closes. Look to the green color, which indicates the neural crest, becomes detached on each side of the neural tube. Okay, this is another video, very nice, showing how the neural groove and the neural crest cells are formed. There's the neural groove, and this thickening on each side is the neural crest. Now there is more deepening of the neural groove. Yes, it will close to form the neural tube, and there is detachment of the cells of the neural crest to lie on each side of the neural tube. This followed by migration of the cells of the neural crest to form different tissues in our body. Development of the flexures of the brain. At first, the neural tube was straight, then followed by development of 3 flexures. What are those? The first flexure is called cephalic flexure. It occurs in the midbrain region. The second flexure is called cervical flexure. It occurs between the hindbrain and the spinal cord. The third flexure occurs in the opposite direction, which is called pontine flexure. It occurs between the pons and myelencephalon. And we will see them in a nice short video. I did it in the next slide. Development of the flexures of the neural tube. At first, the neural tube was straight like this. This is followed by formation of 3 flexures. 1, 2, 3. They are the cephalic flexure, pontine flexure, and cervical flexure. The first flexure to appear is the cephalic flexure. It occurs in the region of the midbrain, so it will be like this. The second flexure is called cervical flexure. It occurs between the hindbrain and the spinal cord, so it will be like this. The third flexure is called the pontine flexure. It occurs between the pons and the myelencephalon, but it will be in the opposite direction, like this. So, this is the final shape of the neural tube. What are the derivatives of the neural tube? Actually, we have to know 3 types of derivatives all over. Derivatives of the neural tube, derivatives of the neural canal, and the derivatives of the neural crest cells. Now we will start with derivatives of the neural tube. At first, the neural tube was straight structure like this. Later on, some changes occurs in the neural tube. What are those? Part of the neural tube lies in the head region, and the other part lies in the trunk. Concerning the head region, 2 constrictions occurs in the neural tube in the head region. They divide the neural tube in the head region into 3 parts, indicated by those 3 colors. I did it by specific colors. The upper vesicle, or the upper part, forms what's called forebrain or prosencephalon, which divides into diencephalon and 2 telencephalic vesicles, which will form the cerebral hemispheres. The next part is called midbrain or mesencephalon, which is this in red color. It gives the midbrain. The third part is called hindbrain or rhombencephalon. It gives rise to the pons, cerebellum, and medulla. Pons, cerebellum, and medulla. The remaining part of the neural tube, which lies in the trunk, it will form the spinal cord. The second type of derivatives is the derivatives of the neural canal. Neural canal, it is the inner space, which is this. It gives the following structures. The two lateral ventricles that are present in the telencephalon. The third ventricle that present in the diencephalon. The fourth ventricle that lies in the hindbrain. And finally, the central canal of the spinal cord. Now we have reached the third type of derivatives, which is the derivatives of the neural crest cells. As we see here, the neural crest cells, it is the second part of the surface epithelium, which becomes detached and lie on each side of the neural tube. What occurs to these neural crest cells? They undergo a process of migration. The term migration is a very important term in the field of embryology. It means movement of structures away from their original site of development to reach new sites. Why? To perform new function. Actually, it gives rise to a large number of derivatives. You have to know them well. What are the derivatives of the neural crest cells? Number one, group of ganglia, which are cranial ganglia, spinal, and autonomic ganglia. Suprarenal medulla. Medulla, not cortex. Suprarenal medulla. Schwann cells, which is the cells that cover the neurons and form the myelin sheath. Melanocytes, the pigment cells in the skin. Odontoblasts of the teeth. Yes, two coverings of the brain, which are the pia and arachnoid matter. Always remember that the dura mater is not included. Carotid body, which is a sensory organ. Ganglionic cell layer of the retina. Yes, there's a nice picture showing the different derivatives of the neural crest cells. Another picture showing the formation and derivatives of the neural crest cells as they do a process of migration. Now we'll talk about development of the spinal cord. There are some changes that occurs in the part of the neural tube that form the future spinal cord. What are these changes? As we see here, the inner lining of the neural tube is called ependymal layer. this layer proliferates, forming an outer zone outside it, which is called mantle cell layer. So, the mantle cell layer forms an intermediate zone, and it will form the future gray matter. What else? Proliferation of the cells of the mantle cell layer will form another layer, which will be the outermost at the margins of the neural tube. That's why it is called marginal cell layer. Yes, myelination occurs in this marginal layer, forming the white matter of the spinal cord. Now, the net result is that we have 3 layers in the spinal cord. The innermost one, which is this one, is called ependymal layer that proliferates to form an intermediate zone of cells, which is called mantle cell layer, that proliferates to form an outermost marginal layer, which is actually called the marginal cell layer. Another changes that occur in the neural tube. What is this? From each side, we have a deep sulcus or groove on the sides of the neural tube. This sulcus is called sulcus limitans. Yes, this sulcus limitans divides the neural tube into a dorsal part, which is called roof plate, and a ventral part, which is called floor plate. What are the other results of the sulcus limitans? Again, have a look on the site of the sulcus limitans. Yes, the sulcus limitans divides the cells of the mantle cell layer, which is this, into a dorsal part, which is called alar lamina, which is pure sensory. It will be like this. And a ventral part, which is called basal lamina, which is pure motor. Both of them constitute the gray matter of the spinal cord. Formation of sulcus limitans and its effects on the development of the neural tube. This is the cylindrical shape of the neural tube. On each side, this duplicated line indicates what's called sulcus limitans. A deep groove occurs on each side at this site of sulcus limitans. It divides the neural tube into a dorsal part, which is called roof plate, and a ventral part, which is called floor plate. In the same time, this sulcus limitans divides the cells of the mantle cell layer into posterior part, which is called alar lamina, which is pure sensory, and a ventral part, which is called basal lamina, which is pure motor. Again, this is the cylindrical shape of the neural tube. This is sulcus limitans. This is roof plate, and this is floor plate. At the region of the sulcus limitans, this is the basal lamina anteriorly and the dorsal lamina posteriorly. congenital anomalies. Before discussing congenital anomalies, we want to ask ourselves how the neural arch of each vertebra is formed. How the vertebral foramen is formed. We will see. This is a vertebra. This is the body of the vertebra. Two bony projections or processes appear from the dorsolateral part of the body of the vertebra like this. Further growth of these bony processes till they meet each other in the midline with the result of formation of the vertebral foramen, and this is called the neural arch. Always remember that the vertebral foramen is enclosing the spinal cord. What occurs if these processes don't fuse with each other in the midline? If this occurs, so this is called spina bifida. Spina bifida. And actually, it has a group of varieties. These are the different varieties of spina bifida. We will discuss them now. But as a general rule, spina bifida, the cause is failure of development of the spines and neural arch of one or more vertebrae. It has two main types, either a manifest type, we can see it, or a hidden type, which is called occult. So we have two main types. The first type is called spina bifida occulta. I can't see the defect on the outer surface of the body. And a manifest type, which is called spina bifida cystica, it gives us a cystic lesion on the outer surface of the body. Now I'll start with the first type, which is spina bifida occulta. Actually, there is a defect in the neural arch, but I can see it. It is covered by skin. But actually, this skin has a specific feature. Usually, this area is covered by a tuft of hair. It will be like this. This is spina bifida occulta, manifested just only by a tuft of hair overlying the region of the defect. So, the spina bifida occulta, this patient appears to be normal. Maybe it doesn't complain of anything or not diagnosed all over his life. But if you find a tuft of hair overlying the lower part of the back in a specific localized region, if you do an X-ray, there will be spina bifida occulta. The second category is called spina bifida cystica, and actually it has different degrees. We'll start by the first degree, which is called meningocele, which is that type. Here there is just protrusion of the meninges. I just find a cystic lesion in the lower back, in the lower part of the back of the newly born baby. It will be like this. The second type of, or the second variety of spina bifida cystica is called meningomyelocele, which is this type. Here, there is protrusion of the meninges together with neural elements. The third variety of spina bifida cystica is called myelocele or rhexisches. In this type, there is exposure of the neural tissue itself on the outer surface of the back of the baby. The fourth and last type of spina bifida cystica is called syringomyelocele. In this type, it is the same as meningomyelocele, but in addition, the central canal is widened, dilated in the region of the anomaly or of the lesion. Now we have reached development of medulla oblongata. What's the developmental origin of the medulla oblongata? Yes, look to the region of the hindbrain and you'll find that it is developed from the myelencephalon, which is a part of the rhombencephalon, which is the hindbrain. Do you remember the three layers present in the neural tube? The inner one, which was the ependymal layer, intermediate one, which is called the mantle cell layer, and the outer one, which is the marginal cell layer. Now I'll talk about the mantle cell layer, which is the middle one. At first, the region of the roof plate is stretched from side to side, like this. It is stretched from side to side, forming the roof of the developing fourth ventricle. In the same time, it becomes invaginated by biamatter, forming what's called tela choroidea, which is a part of the structure of the choroid plexus of the fourth ventricle. As a result of stretching of the roof plate, yes, look here to the basal lamina and alar lamina, which are the two components of the mantle cell layer. As a result of stretch of the roof plate, the alar lamina becomes lateral to basal lamina. It's very important. And both of them are separated by sulcus limitans that differentiate between sensory and motor nuclei present in the medulla. These are the functional components of the cranial nerves present in the medulla. This is the region of the sulcus limitans. And this is the basal lamina with the resultant group of motor nuclei. And this is the dorsal lamina, which is formed of a group of sensory nuclei. Another diagram with the same idea. Now we will revise the functional classification of the cranial nerves to correlate them with those present in the medulla oblongata. Generally, we classify the cranial nerves functionally into either sensory or motor. Now, I'll start by the motor element, which are the efferents. They are further subdivided into somatic and visceral. Somatic, it is just one type. It's called general somatic efferent, while the visceral has two types. Either general, it is called general visceral efferent, it is formed of parasympathetic, and special, which are special visceral efferent, and we'll see now what's meant by each item. I start by the general somatic efferent. It is motor to the skeletal muscles derived from somites. Example, muscles of the tongue, orbit, and so on. They are present in the cranial nervous number 3, 4, 6, and 12. So, they are limited to the tongue and extraocular muscles. What about the visceral? I start by general visceral efferent. It is those containing parasympathetic fibers. They supply the smooth muscles and the endocrine glands. They are present in number 3, oculomotor, 7, facial, 9, glossopharyngeal, and 10, vagus. If you read them in the opposite direction, they form 1973. I think this number is very famous for all Egyptian people. What about the special visceral efferent? Oh, it means motor supply to the muscles derived from branchial arches. It is present in cranial nervous number 5, 7, 9, and 10. What about the sensory component or afferents in the cranial nerves? Yes, we subdivide them into somatic and visceral, and each of them is further subdivided into general and special for somatic, and general and special for visceral. General somatic afferents. It carries sensation from the skin, muscles, and joints, and it is present in cranial nerve number 5, trigeminal. What about special somatic afferents? Oh, it carries two types of sensations, which are vision and hearing. So, from the eye and the ear, always remember both of them start by the letter E. So, it is present in cranial nerves number 2 and 8. General visceral afferents. This type of sensation, it receives sensation from the viscera. It is carried through cranial nervous number 7, 9, and 10. Finally, what about special visceral afferents? It receives sensation from two sides, which are the taste and smell. So it is present in cranial nervous number 1, 7, 9, and 10. Always remember that 7, 9, and 10, they always run with each other. This is the functional classification of the cranial nerves. You can revise it into this nice diagram also, including the distribution of the nuclei into the different parts of the brain stem. Now we'll start our multiple choice questions. The first question, which of the following is a derivative of the hind brain? Pons, thalamus, diencephalon, or midbrain? Yes, think. Pons. Excellent. What is the embryological origin of the cerebral hemispheres? Diencephalon or telencephalon or myencephalon, rhombencephalon or metencephalon? Cephalon. So yes, telencephalon. Excellent. The following are derivatives of the neural crystals except sensory ganglia, cortex of the adrenal gland. Of course, this is because it gives a suprarenal medulla, not cortex. Which of the following is a derivative of the neural crystals? suprarenal cortex، تاني برضه cortex، ولا melanocytes، ولا dura mater، pigment cell layer of the retina، ولا olfactory cells؟ طبعا اكيد هي مين؟ الميلانوسايتس. يلا وصلنا لمرحله ال practical questions. Identify the pointed opening. This is the neural tube. Its upper or cephalic opening is called anterior neuropore. Identify this congenital anomaly. Very nice question. You can take your time. Yes, meningocele. Another question. Identify this congenital anomaly. Oh, there is no protrusion. There is intact skin, but there is a defect in the neural arch. So it is called spina bifida occulta. Identify this congenital anomaly. Oh, here there is meningocele with neural element. So it is called meningomyelocele. Identify the pointed structure. Of course, neural tube. Identify the pointed structure. Yes, excellent. There is neural groove. Now we have reached the interactive questions. I have prepared two clinical problems. First one. A mother noticed that her newly born baby has an abnormal tuft of hair in the lower part of his back. What's your provisional diagnosis and how to confirm? It could be noticed this is a young baby and this is an older boy. It could be noticed at any time all over our life. What's your provisional diagnosis and how to confirm? An obstetrician, immediately after doing a cesarean section, he noticed a midline cystic swelling in the lower part of the back of the baby. What's your provisional diagnosis and how to confirm? Yes, I'm waiting for your answers. We can contact any way of those places.